In this video, we are going to learn about the electron microscope. By the end of this session, you should be able to identify the need for electron microscopes, describe the principles of transmission and scanning electron microscopes, and compare transmission and scanning electron microscopes. Before we get started, it is important that you understand some microscope definitions. So what I would like you to do is pause the video and have a go at writing out the definitions for the words below. Okay, so you should have got, for an image, it's the appearance of an object or material when viewed under a microscope. So it's how you see it when you're looking down a microscope. An object is a material itself that is put under a microscope. The resolution is the minimum distance apart that two objects can be in order for them to appear as separate items. And magnification is how many times bigger the image is when compared to an object. You can work out magnification using the magnification equation, where magnification equals image size divided by actual size. However, we will go through that properly in another video. So what is an electron microscope, and why do I need one? Light microscopes have a poor resolution as a result of a relatively long wavelength of light. So in light microscopes, it is difficult to distinguish between two different objects that are very close together. In the 1930s, a microscope was developed that used a beam of electrons instead of light, and this was called an electron microscope. It has two advantages. Firstly, the electron beam has a very short wavelength, so it has a very high resolving power. And as electrons are negatively charged, the beam can be focused using electromagnets. The best electron microscopes can resolve between two objects that are 0.1 nanometers apart, and that is 2,000 times better than a light microscope. There are two types of electron microscope. You have your transmission electron microscope and your scanning electron microscope. A transmission electron microscope has a resolving power of 0.1 nanometers, whereas your scanning electron microscope has a resolving power of 20 nanometers. The difference in these is due to the different ways they work, and that's what we're going to look through now. So how does a transmission electron microscope work? Firstly, at the top of the microscope, you have an electron gun which produces a beam of electrons. The beam is focused onto the specimen by a condenser electromagnet. In the picture, these are called condenser lenses, but rather than being made of glass, they are made of electromagnets. The beam passes through the specimen, which must be very thin. So to allow electrons to pass through the specimen, it must be very thin. Dense parts of the specimen absorb electrons and these appear dark, whereas less dense parts of the specimen allow electrons to pass through. So a dense part of the specimen could be, for example, your nucleolus, whereas a less dense part of your specimen could be the cytoplasm. Electrons that pass through bleach the fluorescent screen, which is where the electron beam ends up and shines on. This screen can then be photographed, and then this creates your photomicrograph, which is the image produced by a transmission electron microscope. Transmission electron microscopes produce flat 2D images because they pass through that specimen and they only see on one plane. The image below shows two mitochondria that have been photographed using a transmission electron microscope. You can see here that the mitochondria, rather than being their normal oblong shape, are round, and this is because the mitochondria and your cell is 3D, so it depends on the plane that you cut it through. And here you have cut across rather than along, and you get round mitochondria. But you can tell they are mitochondria due to the infoldings of the inner membrane. And if you want to know more about the structure of mitochondria, there is a video on this. So the resolving power of a transmission electron microscope is 0.1 nanometers in theory. However, in practice, this cannot always be achieved. This is because you have difficulties preparing the specimen, and this limits the resolution that can be achieved. Also, to achieve a resolution, you need a higher energy electron beam, and this may destroy your specimen. So it is a trade-off, and generally, resolving power has to be reduced in order to be able to see your specimen. There are other limitations on transmission electron microscopes as well. For example, the whole system must be in a vaccine, and therefore you cannot observe living specimens, so it must be dead. A complex staining process is required, and the image is not in colour. The staining process, rather than using dyes, is using things like heavy metals to absorb the electrons. Therefore, it's not going to produce a colour image, and it takes a lot of skill to be able to stain a specimen for an electron microscope. The specimen as well is even hard to prepare for a transmission electron microscope because it must be very thin. And the image itself may contain artifacts. Artifacts are things that should not be there. They are things that have been produced in the development of your specimen and the staining process, and sometimes they look like structures and they can be recorded as structures, but they are not. So you must be aware for artifacts in your photomicrographs. You will be expected to identify cell organelles on a photomicrograph. So here is your opportunity. I would like you to pause the video and have a go at identifying the organelles that I have identified for you there with the arrows. I will go through the answers in a moment. So pause the video and have a go. Okay, the first one is the plasma membrane. It's relatively easy. It's the one that goes all the way around the edge. The next one's pointed to cytoplasm. As you can see, a cell is actually incredibly full of organelles, but here you can see some bleached areas of not very dense areas, which is where your cytoplasm is. The next one, you've got your Golgi body. If you look closely, you can see the folded membrane and you can see the vesicles where proteins are being packaged and modified. The next label is pointing to a mitochondria, but it's specifically pointing to these infoldings of the mitochondrial inner membrane, which are called the cristae. It's important to be able to identify different parts of the nucleus from a photomicrograph. So here you can see it is pointing to chromatin, that genetic information in your nucleus. Your dense part is your nucleolus, which is where your ribosomes are synthesized and assembled. And then you also have your nucleus, so that's your overarching label. The next one, you have secretory granules. As it is a pancreas cell, they have many secretory granules which will secrete proteins, maybe hormones such as insulin outside of the cell. You have mitochondria there, and you can see it's a slightly rounded shape because of the way it has been cut. And then you've got endoplasmic reticulum. At the edge there, you've got your smooth endoplasmic reticulum. If you look around your nucleus, you can see a darker version of your endoplasmic reticulum. This is your rough endoplasmic reticulum, and it looks darker because you have ribosomes dotted around the edge of it. So the other type of electron microscope we're going to talk about is the scanning electron microscope. The scanning electron microscope starts very similar to the transmission electron microscope, where your electrons are put into your microscope by an electron gun at the top. The beam is then focused onto the specimen by a condenser and objective electromagnets. So they're called lenses again, but rather than glass, they are made out of electromagnets. Rather than the beam penetrating the sample, it passes back and forth across a portion of the specimen in a regular pattern. This scatters electrons, and the pattern of this scattering depends on the contours of the specimen's surface. So, what happens is a computer will analyse a pattern of your scattered electrons and secondary electrons. Secondary electrons are electrons from the sample itself that have been ionised, and they leave the specimen due to increased energy after they have had those electrons fired at them. From this, the computer will build a 3D image of your specimen. So scanning electron microscopes have a lower resolving power than transmission electron microscopes, but produce 3D images, and scanning electron microscopes are generally used to look at the surface of an object. The picture below shows the surface of a tapeworm that can be found in a mammalian intestine. Scanning electron microscopes have the same limitations as transmission electron microscopes. The whole system must be in the vacuum, and therefore you cannot observe living specimens. There is also a very complex staining process, and the image is not in colour. The image may also contain artifacts due to that complex staining process. However, as electrons do not penetrate the specimen, it does not need to be thin, and therefore you can put things in like the whole head of a tapeworm. So you should now know the need for electron microscopes and be able to identify that. You should be able to describe the principles of transmission and scanning electron microscopes, and you should be able to compare transmission and scanning electron microscopes.